Yes, students, so now we are going to study about cilia and flagella, right? First, we will learn the differences of cilia and flagella, right? So, if we will study the difference of cilia and flagella, right? The first difference is cilia are smaller in size, right? And flagella are larger in size. The size of cilia is approximately 5 to 10 micrometers and the size of flagella is approximately 150 micrometers. So cilia are smaller in size and flagella are larger in size. Cilia are numerous, or you can say many in number, right? So a cell will possess numerous cilia, right? Their number is many, and flagella is only one, or there can be two flagellas, right? So these are numerous in number, and this is only flagella is only one, right? The beating of the cilia is synchronous and it is a pendular type of movement. There is pendular movement, right? So cilia beat synchronously and pendular movement. Flagella is having independent movement, right? They are having independent movement. These cilia are of two types, right? Those two types of cilia are Chynosilia. This you have to study later on in detail. Chynosilia are involved in movement, which means those are motile cilia. And the second type of cilia are Stereocelia. Those cilia are non-motile cilia, right? So there are two types of cilia, right? Kyno word is for kinetic movements. So the chynocilia are the type of cilia which will move, whereas stereocilia are also the cilia, but those are non-motile cilia. Those are static cilia, right? Flagella is always motile, right? And there are two types of flagella also, right? Those are whiplash type of flagella, and second is tinsel type of flagella, right? So first let me draw the structure of cilia and flagella. Right? Cilia are numerous and present all over the body. Right? Say for example amoeba or paramecium. Say this is a paramecium. Right? This is a paramecium. Right? So cilia are small structures and those are many in number, numerous in number and present all over the body, right? So cilia are present all over the body. So these are cilia, right? Whereas flagella will be single, right? Say for example in case of sperms, right? In case of sperms, there is a head piece, there is a neck piece, there is a middle piece, and then there will be a tail-like structure. So this is flagella. Flagella is always single, whereas cilia will be numerous in in number and present all over the body. Now, there are two types of flagella, whiplash type and tinsel type. So, if we will draw the flagella, say this is one flagella, it is a single flagella, right? This flagella does not have hair-like or bristle-like structures on its surface. Such flagella is whiplash flagella or naked flagella, right? Whereas the flagellas, this is also a flagella, but this flagella is having the hair-like structures on its surface, right? Those are bristle-like structures. Such flagella is called as tinsel flagella. So, whiplash flagella is naked flagella, whereas tinsel flagella is having bristle-like structure on its surface, right? Examples where cilia are present. Where are cilias present? So, cilias will be present in the nephrons. Nephrons are the part of the kidney, right? Those are the structural and functional unit of kidney. Nephrons possesses cilia. Fallopian tube possesses cilia. Trachea possesses cilia. These are the examples where cilia is present. And which type of cilia we are talking of? This structures possesses Chynosilia, right? Stereocelia is present in case of epididymis and sensory parts, right? This possesses stereocelia, right? So what is it? This cilia will cause the movement of any fluid around it, right? So it causes movement of fluid or structure around it, whereas flagella will cause the movement of cell itself, right? So what is it? We will study it right now. First, remember this much of things, right? So we are talking of differences of cilia and flagella. Cilia are smaller in size, 5 to 10 micrometers. Flagella are larger in size, 150 micrometers. Those are numerous, many in number and present all over the body surface, right? We have drawn the diagram of a paramecium, right? Whereas flagella is only single or two flagellas are present. Cilia, the beating or the movement of the cilia is synchronous movement and it is a pendular like movement. Whereas the flagella beats independently. It is having an independent movement. Two types of cilia are there, stereocilia and chynosilia. Stereocilia are static cilia, which means this cilia do not beat, they do not move, right? Whereas such cilia present, epididymis, which is a part of male reproductive system, and the sensory parts of ear or the sensory parts of eye or the sensory part of the nose will have stereocilia. Chynosilia are involved in the movement. Those are motile cilia. Where are chynosilia present? Those are present in the nephrons of the kidney, in the fallopian tube, right? Or in the trachea. Flagella are of two types, whiplash and tinsel type of flagella. Flagella will always be motile. The structure of eukaryotic flagella is different from that of prokaryotic flagella. Now two types, whiplash, it is a naked flagella. It does not have the hair or bristle-like substances present on its surface. Whereas tinsel type of flagella is possessing the bristles or hair-like structure on its surface. So these are the two types of flagella. whiplash and tensile type. Cilia is involved in the movement of the fluid or structure around it, whereas flagella is involved in the independent movement of the cell. What is it we will study, right? So cilia is involved in the movement of the substance around it, right? Say for example, as I told, cilia is present in the fallopian tube, right? So say this is ovary and this is the fallopian tube, right? The ovary, this is ovary and this is fallopian tube, right? Now the cells of the fallopian tube, these are the cells of fallopian tube. This cells of fallopian tube will have cilia. As I said, cilia are numerous in number. So these are the cilia present on the cells of the fallopian tube. Now ovary is releasing ovum, right? So ovary, say this is the ovum which is released. This ovum enters into the fallopian tube. Now this ovum has to move through the fallopian tube to reach towards the uterus. Now how is this ovum going to move? Who is going to help it? Then this Chynosilia are going to help it. Chynosilia will beat synchronously and continuously so that the ovum is transported towards the uterus. Similarly, it is present in trachea, right? So again, trachea is a pipe-like structure. Say this is trachea and these are the cells of trachea. Now the cells of trachea also possesses cilia, right? Those are also chynosilia. So these are the chynosilia present on the cells of the trachea, right? Now say there is production of mucus, right? There are goblet cells also present and that goblet cells have produced mucus. This is the mucus and the cuff, right? Now this mucus which is secreted in the trachea should not go into the lungs, otherwise it will damage the lungs. Then where should it go? It should be expelled out either through mouth or through nose. So this mucus is transported upwards towards the mouth or nose because of the beating movement of this cilia, right? So the cilia will beat to transport the mucus up. So cilia is involved in movement, but it is not moving the cell. It is moving the fluid or the structure which is present surrounding it. Flagella is involved in the movement of the cell itself, right? Say for example, where is flagella present? Example, sperm. Sperm have flagella. So this flagella of the sperm is involved. Say this is the nucleus of the sperm, is involved in the movement of the sperm itself. So it is involved in the movement of the cell itself. Cilia causes the movement of the structure around it, whereas flagella is involved in the movement of the cell itself. Right? Now we are going to study about the structure. The structure of cilia and flagella, internal structure is common. Right? We are going to study it. So now we are going to study the structure of cilia and flagella. Yes. So the first thing is it is covered by a plasma membrane sheath and the center core is called as axoneme, right? And it is made up of microtubules in the arrangement. That arrangement is called as 9 plus 2 arrangement. The anchoring structure is called as basal body or blepharoblast or kinetostome, and its structure is similar to the structure of centriole. Its structure is similar to the structure of centriole. So what is it? We are studying the internal structure of cilia and flagella. Cilia are numerous and smaller in size. Flagella will be single and larger in size. Right? So whatever it may be, say this is a cell, say this is a cell, and this cell possesses this cilia. These are the cilia. So this cilia will be covered by plasma membrane. It would be covered by a sheath of plasma membrane, and the center core-like structure is called as axoneme. So the core-like structure is called as axoneme. But see, if there is something which is hair-like outgrowth, this cilia and flagella are nothing but hair-like outgrowth. So if hair-like outgrowth is there on the cell, right? Or say for example, we are talking of another cell, right? This is the cell and this is the flagella, right? So if the hair-like outgrowth is there, then there should be some root or something which should anchor it. That anchoring structure is called as basal body, right? So who is anchoring it? It is anchored by basal body. So this structure is nothing but basal body, also called as blepharoblast or kinetostome, and its structure is similar to the structure of centriole. We will study the structure of centriole. So cilia is having actually two parts: the basal body, which is the anchoring structure whose structure is similar to centriole, and this hair-like projection. It is covered by plasma membrane, and the center core is called as axoneme. Right? Now this hair-like projection, we are going to study the structure of this hair-like projection. So the anchoring structure is basal body. Right? So we are going to study the structure of this hair-like outgrowth, that is cilia and flagella. The structure of basal body is similar to centriole, which we are going to study in the next chapter, next lecture. Right? So the internal structure is having 9 plus 2 arrangement. Right? So the arrangement, you have to remember it, very important students. The arrangement is 9 plus 2 arrangement. Right? The arrangement is plus 2 arrangement. Right? Now eukaryotic cilia, or we can say more precisely flagella, is made up of tubulin protein, whereas prokaryotic flagella, the structure of prokaryotic flagella is different, and it is made up of flagellin protein, right? So the structure of eukaryotic and prokaryotic flagella are different, and eukaryotic flagella is made up of tubulin protein, prokaryotic flagella is made up of flagellin protein. The arrangement of eukaryotic flagella is 9 plus 2. Now flagella, so we are talking of eukaryotic flagella. This eukaryotic flagella is made up of microtubules. Its main constitution is microtubules, and these microtubules are made up of tubulin protein. So why we are saying 9 plus 2 arrangement? that we will study, right? So, what is the structure of cilia and flagella, right? So, first, it is covered by a membrane. That membrane is the plasma membrane, right? So, plasma membrane is covering it. In the center, there are two microtubules present, right? And what is the structure of this microtubule? So, this microtubules are this tube-like structures, right? Microtubules are tube-like structures, and these are made up of two types of tubulin protein, right? So, say this is one tubulin protein, this is alpha tubulin, and the other tubulin protein is beta tubulin, right? And this alpha and beta tubulin are arranged alternately with each other, right? So, alpha and beta tubulins are arranged alternately. This is the structure of microtubule. This is microtubule. Microtubule is made up of alternate arrangement of alpha tubulin and beta tubulin proteins. And cilia and flagella are made up of microtubules. Those are pipe-like or tube-like structures. Now, in the center, there are two microtubules present, right? So, let me draw it. This is one microtubule, and this is another microtubule, right? These are the two centrals, C1 and C2. And how many subunits are present in it? Then it is having 13 subunits. 13, right? Similarly, this is also made up of 13 subunits, right? So, in the center, two microtubules are present. These two microtubules are connected with each other by a bridge, right? So, these are the central microtubules. This is the bridge, right? Central bridge which is connecting the two tubules. This is surrounded by a coat, that is proteinaceous coat, right? It is surrounded by proteinaceous coat. So, in the center, two microtubules are present. Now, what is present in the periphery, right? In the periphery, so this 2 stands for central microtubules. In the periphery, there are 9 doublets of microtubules, right? So, in the periphery, there are 9 doublets, which means there will be 2 tubules, right? So, say this is one tubule, it is A tubule, which is complete. And why it is called as complete? Because it is made up of 13 subunits. Whereas the B tubule, this is B tubule, and it is incomplete. Why? Because it is made up of 10 subunits. This B is incomplete, right? So, this is a doublet. This is a doublet. 9 such doublets are present in the periphery. So, I will simply draw it. I am not going to draw the inside tubules, right? So, let me draw it. This is 1, this is A, and this is B. A, B. Again, this is A, and this is B. Again, A, B. A, B. Again, this is A, and this is B. 1, 2, 3, 4, 5. This is 6th one, A, and B. This is seventh one, A and B, 8, A and B, and this is ninth one, A and B. So this is, let me number it, 1, 2, 3, 4, 5, 6, 7, 8 and 9. 9 doublets are present. Right? Now there is a linker. A microtubule of first doublet is linked with the B microtubule of its succeeding doublet by a linker. This linker is called as AB linker. Similarly, this A with B, this A with B, this A with B, likewise those are connected by doublets. Right? Draw it like this. Right? So this A is connected and this A is connected with this. This much is clear, students. Right? So this I'm not, so that the figure is clear. Right? Right? This much is clear, right? So in the center there are two microtubules which are separate, right? And C1 and C2 which are connected by a bridge, right? That is the central bridge, right? Surrounded by a proteinaceous hub, right? In the periphery 9 doublets are present. This 9 doublets, which means there are two microtubules. A is complete, which means it is made up of 13 tubulin proteins. B is incomplete, it is made up of 10 and 3 tubulin proteins it is sharing with A. Right? So such 9 peripheral doublets are present. A tubule is connected with the B tubule of the other doublet by a linker. That is AB linker. Then one more thing is present, right? There are dianein arms which are present, right? So this A tubules will have this arms, right? There are arms which are called as dianein arms. So these are the dianein arms. These are inner dianein arms and these are outer dianein arms. So these are dianein arms. Those are made up of dianein protein. And very important, students, that this dianein arm is functioning as an ATPase, which means it is breaking down ATP into ADP. Energy is released and this energy is utilized for the beating movement of the cilia. One more thing, all these peripheral doublets are connected with the center by the radial spokes, right? So those are connected with the center by the radial spokes. This much is clear. So this is the structure of cilia and flagella. 9 peripheral doublets are present. So you can say that 9 into 2. So 18 microtubules are present in the periphery and 2 in the center. Right? So if the question is asked that how many microtubules are present in total in the cilia or flagella, the answer should be 20. Right? 9 doublets, so 18 in the periphery and 2 in the center. So centrally 2 microtubules are present connected by bridge, periphery 9 9 doublets are present. A is connected with B by the linker, that is AB linker. A tubule is having arms. Those arms are called as dianein arms. Inner dianein arm and outer dianein arm. Very important, dianein arm is functioning as a motor. Right? It is functioning as a motor or ATP as enzyme, which means it is utilizing energy for the beating movement of the cilia. Right? This much is clear. Right? So cilia and flagellas are having its own movements and dianein is helping it. The structure is 9 plus 2, 2 central and 9 peripheral doublets. What is it? What are the structures? These are microtubules. Microtubules are tube-like structures. What are it made up of? It is made up of tubulin proteins, alpha and beta, which are arranged alternately with each other. Right? So this is the structure of cilia and flagella, that is hair-like outgrowth. The anchoring structure is basal body. The structure of basal body is similar to centriole, the structure of which we are going to study in our next lecture.